Hello, everybody. It's Mr. LaChance. I hope you guys are happy, healthy, and getting outside into this beautiful spring weather we've got going on. This video is to help you better understand the concepts behind the case study we're going to work on this week. You'll also notice there are two total videos in this assignment. This one, will let you know terms and things you need to know before you get started. And then the next one will be an example of navigating the directions, because if you've opened up the document, you've already realized that, wow, there's a lot of letters there. The directions are pretty intense in regards to navigating this database we're going to use. So I've got them typed out for you, and I'll have a demonstration in that other video. All right, let's get started. Okay, things to know as we get into this case study a little bit. So we're trying to figure out, are bats birds? And many of you might already know the answer to that question, but we're going to prove it using information from a protein database. So beginning terms, a species, we're going to compare 6 different species to determine relatedness. But generally speaking, species are a group of genetically similar organisms that are capable of interbreeding and producing viable, fertile offspring. So what we've been learning about in evolution and also thinking back to genetics, when a speciation event happens, meaning one group splits into two, basically what happens is the two gene pools for each of the groups become isolated, and they're no longer mixing together like they did in the past. And so as those two genomes diverge and they become isolated, they're each going to accrue their own set of random mutations, which will cause differences in the genome. And also, due to selective pressures of the environment, the genome will be sculpted over time. So the idea is, once two species split apart, their genomes become more and more different as time elapses. We've also been learning about phylogenetic trees. Phylogenetic trees are branching diagrams that show the relatedness of various species or groups of species. And we know that the more recently two species branched apart, and the more branches they share on the tree, then the more closely related they are. So if we were to compare the sac fungi and the club fungi, and if we were to look at their branches and go backwards in time, we notice that they diverged more recently than the other groups of fungus have. And so that means that the sac fungi and the club fungi, out of all these 5 groups, are the most closely related, and they share a more recent common ancestor. Now, if we look at the sac fungi compared to the chytrids, and we travel back down the branches through time, we'll notice that those 2 branches divided a long time ago, at least compared to the other ones. So they share a less recent common ancestor. So what is a common ancestor? Well, it's hard to know exactly what they looked like, but by looking at a tree, we can make some general assumptions. If we look at humans and chimpanzees and scroll back to the last branch in which those 2 groups were connected, you'll see the yellow dot. So the yellow dot is the population that was around right before the human lineage and the chimp lineage diverged. And so that yellow dot represents the last common ancestor that humans and chimpanzees have shared. Now, once again, what did that look like exactly? It's hard to say. Was it a chimpanzee? No. Was it a human? No. It had qualities shared by both of those things to an extent, but we never know exactly what a common ancestor looks like, but we can just assume that because humans and chimpanzees have such similar genomes, they diverged relatively recently in their evolutionary history. So this brings about a big misconception that's aligned with evolution. Many people think that we evolved from chimpanzees, when really we share a common ancestor with chimpanzees. Okay, so once again, for this case study, we're going to compare protein structure across 6 different species to try and figure out if bats are birds. So to understand why we're doing this, you need to recall what we labeled the central dogma of genetics, which is the picture on the right. It's the idea that the information encoded in DNA eventually leads to protein structure. So as we're comparing 2 species to determine relatedness, we could look at their genomes And knowing that the genome is closely related to protein structure, we could also look at a common protein that both those species have, compare its structure, meaning the sequence of amino acids, and then determine relatedness through that. So for this lab specifically, we're going to look at a protein called cytochrome B, and it's a protein that'll be found in all the species we're going to analyze because it's found in the mitochondria of all eukaryotic cells. And bats, birds, and mammals are, of course, eukaryotic. Okay, so here's a picture of the database we're going to use. And here in a minute, I'll show you how to navigate it. But first, a few terms present in the database that we need to understand. So when you see the term length in the database, that is referring to how long the protein is. And so by length, we're talking about how many specific amino acids make up that protein. And so for cytochrome B, there are roughly 379 or 380 bits to the length. So that just means that there are 370-ish or 80-ish amino acids that make up that specific protein. So that's length. There's an align feature with this database we're going to use. And when we align proteins, basically the program, the database, will look at the sequence of amino acids for both of the proteins or all the proteins involved and compare similarities and differences. So of course, if a protein structure is very similar across two species, then they're closely related. If it's not so similar across two species, then they're distantly related. But we're going to compare proteins of bats, birds, and mammals to see who's most closely related to who. Okay, the last term we'll talk about, identity. So in the protein database, when we align two proteins from two or more species and we compare them, the program will kick out what's called an identity. The identity is expressed as a %. And basically what it means is if we were to compare those two proteins, just like the database did, we would notice that percentage of amino acids in the exact same spot. So let's say we compare two species and it kicks out an 87% identity. What that means is 87% of the amino acids that make up those two proteins are exactly the same in exactly the same order. So of course, a higher number means that the species are more closely related, and a lower number means there's less similarities in their genomes and their proteins, and so they're more distantly related. Okay, so this is the end of this first video, which is once again just to kind of define the introductory terms and give you some background knowledge. The next video you'll see in this assignment portal is a demonstration of how to navigate the directions. So please open that up now.